In the previous video, I discussed mitosis and meiosis. Now, I will go through this in detail. So, first here, M phase. Actually, M phase means nothing but mitotic cell division. In the previous video, we discussed the G1, S and G2 phases. Okay, now in detail, let us learn. So, M phase is nothing but mitotic cell division, and this is the duplication division. Okay. Meiosis is reduction division. So here mitotic cell division is having 4 stages: prophase, metaphase, anaphase and telophase. This we say as PMAT. Okay, PMAT. Like that you can remember: prophase, metaphase, anaphase and telophase. Okay, and this is followed by cytokinesis. That is division of cytoplasm. Now here Potu, Narasimha Rao and Johnson, these two are the scientists, and they for the first time they revealed the relation between these 4 phases. Actually, before that, the scientists could not understand what is the relation between this PMAT. But here, this is recent development. Mr. Narasimha Rao and Johnson, these 2 scientists have revealed this, the relation between these 4 phases, and they for the first time they revealed that these stages are there: G1, S and G2 phases. Actually, this is it's very difficult to demarcate these phases. So between these phases, these subphases are there. So for the first time they observed these subphases. And how did they see? Because chromosomes cannot be seen by the microscope. So these subphases for the first time they revealed, and how did they reveal? That is under the controlled unidirectional method and controlled way. So sequential stages they have observed very slowly, and they in a single direction they did these experiments, and they under the controlled way. So like that they observed these subphases. Okay, and like this cell cycle is progressed, okay, under these conditions. So this has helped very much in further cell to know about the cell divisions. So this is treated as a milestone in the cell cycle studies. Okay, now here actually some chemical signals also they used for this, that we call as cell fusion technique. Okay, cell fusion technique, that is combination of 2 cells in experimental conditions. Okay, then they clearly demarcated the nucleus in the cytoplasm. So the division in the nucleus and division in the cytoplasm, that was very much clear because of this cell fusion techniques. And then stages of mitotic cell division. So stages of mitotic cell division. So actually this is mainly of two divisions. First one is karyokinesis, division of nucleus, and cytokinesis, division of cytoplasm. So these PMAT, let us learn with the help of the textbook, or if you open your textbooks, I'll give you some description, brief description regarding these 4 stages. Open your textbooks. Page number 136. All of you open. Now look into the textbook. First one is prophase. In this prophase, actually chromosome will condense and it will be coiled. Then this chromosomes can be visible with the help of microscope, and nucleoli becomes very small. And the second thing is chromosome will split lengthwise, and each split part is called as chromatid, and these 2 chromatids will be connected by the centromeres, and gradually the nuclear membrane will be disappeared. Then centrosome containing rod-like centrioles divide and form ends of spindles. So spindle fiber formation will be taken place here, and at the middle centromere will be there, and that is connected with rod-like centrioles, and this will be attached with some spindle-like fibers. Then this can be seen only in animal cell but not in the plant cell. Okay, then next one, the important point here is chromosomes will not be paired. Okay, but in case of meiosis, chromosome pairing will be seen because XY chromosomes will be fused together, or XX chromosomes. But here in mitosis, pairing will not be there. And then next one is metaphase, and chromosomes here move towards the equator, that is at the center, and these spindle fibers attached to the centromeres. Just like they will be holding at the center with the help of these fibers. And then these centromeres will split, and the chromatids will be separated towards the poles. then centromeres will split and chromatids will be separated. From the center, they move towards the poles. Then anaphase, third phase, and here the spindle fibers attached to the centromeres contract and pulling to chromatid towards the poles. So, this, moving towards the poles will be done in anaphase. The spindle fibers will be contract. And then telophase, the last stage. Here chromatids elongate, become invisible. Replication at this stage to become chromosomes. So, actually, again, because the two daughter cells will be formed, each daughter cell should have the chromosomes. That's why again chromosome formation will take place here in the telophase, that is the last stage, and nuclear membrane will appear and they form around this daughter nucleus. So two daughter nuclei will be formed, and then cell membrane pinches to form daughter cells or new cell wall material becomes laid down across spindle fiber. So here new cell membrane will be formed and this nucleus will divide into two, and the cytoplasm, that is cytokinesis will start here. Okay, and finally two cells will be formed, two daughter cells will be formed. So this is mitosis. And now look at the board. I discuss the remaining part now. Coming to meiosis, and this is reduction division. Already we discussed this before. And here during the formation of gametes, actually meiosis takes place only during the sexual reproduction. That's why it is reduction division. The chromosome number will be reduced to half, and here it contains first phase and second phase. And first phase will have, actually the parental cell will have two sets of chromosomes. It undergoes two divisions, subsequential divisions it undergoes, but only one, this chromosome will undergo only one division, whereas cell, the cytoplasm will undergo two divisions. So first phase only cell cytoplasm will be divided and chromosomes will not be divided. Only once it will be divided. And then we take the second phase under meiosis, and this is just like the mitosis only. And here at the end of this second phase, four daughter cells will be formed. At the end of meiosis, four daughter cells will be formed, and each daughter cell will have haploid number of chromosomes, that is single, not double. Okay, and here no chromosomal duplication. This you must remember, same as mitosis. Okay, so chromosomal duplication will not be there, and equidistribution of chromosomes will be there, because the chromosomes should be equally distributed to all the four daughter cells. So this will be done at the end of meiosis. And now I will continue this. Yeah, you might have heard about this AIDS disease. Actually, this is a viral infection, and this is also because of not ill healthy way of sexual contact and all. And this AIDS is a viral infection, and actually here to acquire, actually what is this AIDS? Full form I am discussing now. A is acquired, I is immuno, D is deficiency, S is syndrome. Actually acquired immunodeficiency, that means here the immunity system will be disturbed first. So when the immunity is not good, automatically all the diseases will attack. All the problems will be there for the person. So like this AIDS will cause, that will lead to the death of the person, because the immunity system will be disturbed and that will become very weak. Then here in order to create the awareness, some organizations are there, and the government of India, actually the central government has progressed many the programs, and among them Asha, Red Ribbon Express, and these two are important. Actually these are running by Medical and Health Department and Family Health Department. Okay, ASHA means accredited social health activist. Then Red Ribbon Express, these two are the organizations to get to create the awareness among the public. And availability of medicines, actually government is supplying some medicines through this ART centers. Actually ART means here anti-retroviral centers. So here these AIDS patients will get the medicine for the free of cost. Then next one is birth control method, because actually this is very important awareness among the in the society, because why these problems will come. This is because of the illiteracy, poverty, ill health, malnutrition. All these are the factors of this problem to the mother, actually motherhood. So here contraception is among them. Actually this is to control the birth, because early marriages, first of all, early marriages, this early marriage system should be eradicated. Because of this, because the mother is may be physically mature, but mentally the maturation will not be there to bear the baby and all. So that should not be there. So child marriages should be banned first. Okay. So for this, actually, early marriages first of all, then contraception. Actually, the prevention of pregnancy in women by preventing fertilization. So here fertilization is stopped. I mean, that is prevented. Okay. So this is called as contraception, and the device or the drug, whatever we use for this, is called as a contraceptive. And spermicides. Nowadays, spermicides are available, and this is used to kill the sperms in male. That is in the form of pills. Then vasectomy and tubectomy, these two are the surgeries to stop the fertilization. So vasectomy is a small portion of vasa deferens. First, actually, the vasa deferens, a small portion will be cut by surgery, and both the ends are tied. And tubectomy, this is male, and in case of males, and in case of females. So how can you remember? Because generally children will get confused with vasectomy and tubectomy. Tubectomy means actual tubes will be there, fallopian tubules in female reproductive system. So like that you can remember. So tubectomy is in case of females, and vasectomy is in case of male. In case of vasectomy, vasa deferens is cut, whereas in tubectomy, the small portion of fallopian tubule will be cut. Removed by surgery, and both the ends will be tied, so that the fertilization will not be taken place. Okay, so like this, birth can be controlled just to stop the population growth, as well as the healthy motherhood. Okay, now I'll discuss the general things about in the society. So for this, actually, female feticide. See, if the baby is in the fetus, already I told you which stage is called fetus. Okay, so in the fetus, when the fetus is there in the uterus, then many people are going for abortions and all. That is feticide. We call it as feticide. So if the baby is a girl child, then they are going for this feticide. Actually, it should not be there, and the doctors are not allowed to do this feticide and all. So abortion should be stopped, and that should not be there. And the mother is not ready to carry the child or take care of the child. So early marriages or teenage marriages, teenage motherhood, that should be stopped. And then so these are the things. And at the same time, female feticide should be stopped, and ultrascanning, scanning and all, the mothers are going for to know about the sex of the fetus. So it should not be done, the baby, and it should not be done. And the doctors are strictly, many acts, our government also our government also enacted many laws to stop this, know about the sex of the sex determination before the delivery, and that should be stopped. And beyond all these things, actually, every person, every individual should have the awareness of getting married, the age for the actual marriage, and also unhealthy sex contacts and all, and also the social awareness every person should have. So individual thinking and individual participation should be there for the healthy growth of the society. Thank you.